o'clock, so let's get started. My name is Valerie Cotton, and I'm just going to kick off this webinar. Welcome to the webinar on the Kids First and KOMP2 collaboration for precision modeling of pediatric conditions. This slide is just to tell you that you all have been muted upon entry, and we just ask that you remain muted until the question period, just so we can kind of regulate any background noise. However, you can unmute yourself in various ways. If you called in on the phone, *6 will do it. If you called in on the computer, you can unmute yourself under the Quick Start tab. And you can also message me through the chat function on WebEx. I'm recording this webinar so that we will make it available on the Kids First Comments on website after the fact so that you can share with your colleagues. So again, just allow me to introduce this webinar simply by saying that on behalf of the Gabriella Miller Kids First Pediatric Research Program, we're really excited to engage in this collaboration with the Knockout Mouse Phenotyping Project. We really think that this collaboration that we'll tell you more about will accelerate the genetic discovery of pediatric conditions using Kids First data sets. And with that, today, first you will hear from KOMP staff. Steve Murray will give a presentation explaining some background about the KOMP2 program and some details about this specific collaboration. Then James Coulomb from the Kids First Working Group will tell you give a very brief background about Kids First and then tell you how you can nominate variants for this opportunity. And we will take questions at the end. So Steve, let me turn it over to you.
Okay, great. All right, and I will share my screen. Oops, hold on just a second.
Yeah, no worries.
Okay. All right, so can everybody see my screen?
Yeah.
It should be a full screen.
You might want to make it big, though. There you go.
Are you looking at the center notes? Or, anyway, as long as you can see it well, I think I'll get started. Okay, so I'm really pleased to present some context for our program overall and some discussion points about how I think we could interact as a group. I want to, before I start, thank my colleagues, Kent Lloyd, Laurel Nutter, John Stevitt, and Steve Brown for helping contribute to this presentation, both in editing and slides. And as you'll see in a moment, this consortium functions very much as a consortium in a partnership. And I want to thank them for helping put this together. So I'll just dive right in from there. So just to present the Knockout Mouse Phenotyping Program, and our goal is to build the first comprehensive catalog of mammalian gene function, and I'll jump in from here. So I want to start with a little bit of the context and the challenge that we embarked on at the beginning of this project. And it started with the notion that a mammalian of the genome is what we call dark, that function of the genes are unknown for the most part. I think I would say the majority of the genes, are still underrepresented in our general knowledge base. And this was, for many of you who probably saw the recent report and the recent paper from PLOS Biology, further illustrating that we tend to look under the lamppost for things and that we don't actually spread our wings into the rest of the genome particularly effectively. So recognizing that, we recognize that particular challenge. Likewise, the functional consequences of human genetic variation are poorly understood in general. I think this is a challenge that we face both in model organism research, but clearly in human genetics research as well. An area we're beginning to explore, and an area that I think is ripe for further investigation, is the functional analysis of the non-coding genome, which has been thus far quite limited for the most part. And as those of you on the Kids First side are well aware, the number of variants of unknown significance is increasing quite rapidly, but we haven't really met that from an analysis of function, particularly in vivo function. And we believe, and we strongly believe, that the mouse as the premier mammalian system, model system, is critical to dissect and understand the importance of genetic variation. Just to give you some context for the structure of our consortium, We are composed of three different, the KOMP2 program overall is composed of three different consortia. Our colleagues, led by our colleagues at University of California, Davis, with the Center for Phenogenomics in Toronto, along with Charles River and Corey, is the DTCC consortium. A group led by Baylor College of Medicine with contributions from both the Sanger Institute and now, and also the MRC Harwell is the BASH consortium, and then JAX is a singular consortium, I guess, here at the Jackson Laboratory. We together have been funded for 7 years now, and we're in phase 2. In the first phase, we generated and phenotyped. I will update my slide to include the past tense, 2,500 knockout lines for the KOMP2 project. And we're now in phase 2, which has a larger goal of 3,000 knockout lines during this 5-year window. And with our international partners, we have the goal for the completion of the genome. Just so you're aware, this is a trans-NIH effort. It's funded by both the Common Fund and all of the institutes and centers contributed to this program overall. So it's truly an integrated program. A few notes that I wanted to make that are principles that are part of this program overall. They include really a focus on standardization and reproducibility. I will give you some more details of that in a moment in terms of the phenotyping, but one of the fundamentals of our standardization is a uniform C57 black 6N genetic background for all of the strains that we produce in phenotype. As I'll go into a little more detail, we use defined validated alleles. We want our users to understand and know in certain terms what exactly the mutations are that we generate. And then standardized phenotyping protocols. We've spent an enormous amount of time and effort to either standardize our protocols or be deliberate in describing the places where we differ, such that the data is maximally interpretable by the scientific community. We are absolutely committed to rapid data release through a centralized portal at mousephenotype.org. All centers upload their data not only on a regular basis, but on an immediate basis for the most part. And I think this makes us, if not unique, fairly unique overall. And we're also committed, have always been committed to the open availability of mouse resources and other materials through centralized repositories. As I mentioned, I mentioned the KOMP2 program, but we are part of a much larger partnership with centers throughout the world. This is just an illustration of all the different centers and their locations and continents spanning the globe. So, you know, this is a large, audacious goal to knock out every gene in the genome, and in order to do that, we need partners from all over. So, as I mentioned, the goals are to ultimately generate a null mutant for every protein coding gene in the genome. But the key point here is we're also comprehensively phenotyping each mutant to determine developmental, physiological, and biological parameters, and to provide the maximum amount of data for every mouse that we generate. We believe this is critical, provides a critical baseline for exploring gene function, and thus to develop a more profound understanding of the genome landscape and genetic mechanisms. Where it's clearly becoming increasingly important is to enhance our interpretation of human genetic data, including rare and complex diseases and the analysis of large population data sets. It's evident to us that the value of model organism data is very important for this research. And as I said, we're moving more towards a functional annotation of human genetic variation, which I think will be the basis of this collaboration. Just to give you a little taste of some of the impact that we've had just within our consortium and the papers that we've begun to write to describe our discoveries within our own resource. Right here, we started back in 2016 with our paper describing the developmental phenotypes, and I'll come back to this in a moment, the high rate of lethality, or the significant rate of lethality that we found in our mutant lines. But it's been followed by a series of high-profile publications describing either disease relevance or collections of mutants with individual traits. We certainly expect that we will be seeing this continue, but what's really satisfying to us as well is when we see other groups around the world using our data and publishing their own papers with this, and this continues to grow quite rapidly. To give you a snapshot of where we stand today as a consortium, meaning the IMPC overall, I know as somebody who was a major part of the production aspect here at JAX, it's amazing to see that we've made over 12,300 attempts to create a mouse line, that is individual microinjections, or now with CRISPR electroporations, but individual engineering attempts. And this has resulted in a current snapshot of over 7,500 genotype-confirmed knockout lines thus far. And to put this into some context, the genome is estimated anywhere from 19,000 to 20 genes. So we are rapidly approaching half of the entire genome, which is quite exciting from our perspective. And I'll also note, you'll see here that greater than 2,000 of these knockouts have been generated by CRISPR, and I'll come back to that in a moment because that is now our prime mode of operation. Of these, 5,800 have been expanded to the point where phenotyping is complete, and you'll see that our data release 8.0, which is pending or currently live, I actually don't, it's pending very shortly, it will be for 5,500 lines, which is really remarkable. A quarter of the genome we have full phenotyping data for. That accounts for an astounding 61.7 million data points overall and about 369,000 individual images, and you'll see some of them right here on the slide. To step back a second, and I think this is really important for understanding how we generate our knockouts. In phase 1, many of you are probably aware that we used resources that had been generated by the International Knockout Mouse Consortium as cell line resources, and this comprised cell lines of 2 flavors, which were either the knockout first conditional ready multifunctional allele or the velocigene knockout first, or I'm sorry, definitive null knockout approach, which knocked out every coding exon in the genome. And these were our workhorses for the first 5 years, essentially, of the program overall, and so many of our lines were made this way. And the added piece of information here is that this legacy information also contains gene expression information that was enabled by the LAC-Z reporter that was part of the allele. Because of the profound increase in efficiency and reduced costs that CRISPR-mediated mutagenesis provided, in phase 2, we moved to an entirely CRISPR-based exon deletion or small indel or deletion-based approach, primarily because we wanted to go further in the genome and use less resources to do so. So our main goal was to move into the dark genome, and the way we could accomplish this was to make more simplified alleles, but to do more of them and to do them at a remarkably reduced cost. And our general strategy here is to either generate a frame-shifting mutation by deleting an entire exon, which is akin to the approach for the IMPC alleles. However, we do recognize there are many gene models in the genome, either single exon genes or genes for which all the exons are in the same phase, for which we have to use slightly different strategies that still involve a substantial deletion, but we will screen for the creation of a frame shift mutation in these cases as well for these classes of genes. So going forward, all of our nulls will be of this CRISPR-mediated type. I mentioned before, we have developed, in close coordination amongst the entire consortium, a unified set of parameters and tests that contribute to our understanding of a variety of different physiological systems. So you can see a number of different tests are a part of the neurobehavioral system, a number of metabolic and body composition-related tests. We have cardiovascular, reproduction, extensive sensory testing, musculoskeletal, immunology, behavior, I mentioned that at the beginning, and then and also general histopathology and other general clinical assessment parameters as well that can be used to provide deep information about each individual knockout line. In order to make this happen and make sure it's cost effective, we've worked very hard to organize this into a linear pipeline in which individual tests are performed at specific weeks. This this scheme was constructed with the recognition that previous tests can affect subsequent tests, that different tests require anesthetic, et cetera. So all of this has been validated to work in a specific order, and it maximizes the efficiency. We run 7 males and 7 females, and then local controls during the course, wild-type controls during each, through the entire pipeline. And ultimately, the structure here maximizes the amount we can do in a cost-effective manner and extract the most data we can from any given mouse and any given cohort of mice for a given line. The lines are also, the reason I point this second part out is because initially all of the lines were done in an early phase between the ages of 8 and 16 weeks. You can see now we've expanded this for a subset of the lines into what we call our late adult pipeline, which is essentially the same pipeline, but typically starting much later in life. You can see 49 to 70 weeks, depending on the center. Some centers have gone out a little bit further. Some centers have also included additional tests that are non-terminal, of course, during the aging period as well. And both for both the early adult and the late adult pipeline, we have an extensive set of terminal procedures, again, as I said, to maximize the amount of value you can get out of every single mouse that we produce in phenotype. So what are we finding? And one of the really interesting features that we've discovered over the time is not only a very high hit rate, but also evidence of extensive pleiotropy. And we think this is one of the most important things that we can present from the IMPC. Much of the published data necessarily is restricted to the expertise of the investigators who generated the original knockout. So by performing broad-based phenotyping, we capture information in all areas of physiology, not necessarily those dictated by our own individual expertise. So what we found is, this is a little bit old data, obviously it's not the current release, but from 3,500 genes that have sufficient phenotyping complete, which we're defining as greater than or equal to 13 of the phenotyping parameters, because this is an ongoing process. Not every line is sufficiently complete for this definition. But what was really interesting is that we found fully 83%, almost 84% of these lines showed at least one phenotype hit, which is really remarkable given that we are, as I said, diving into the dark genome, but of course confirms our suspicions that evolution is fairly conservative and that genes remain non-pseudogenes for particularly good reason. So, and it also gives us, it's buoying because we know we're testing a really effective range of phenotyping parameters that allows us to detect such a high rate of a high hit rate overall. And just to mine some of the data that's been published, which, as you know, these are data for genes for which we expect something to happen. What is published is that about 89.7% report at least one phenotype, typo there, which is remarkably close to our 83% for genes for which we often know almost nothing. So I think this is valuable information not only for our current data set, but it also tells us going forward we can expect to continue to find a lot of interesting phenotypes. Our hit rate varies significantly by system. Part of this is the depth to which our pipeline is structured for those systems, but other elements here might reflect the number of genes that are important for a given physiological system. The one I will note in particular, our highest hit rate is for mortality/aging, which essentially means mortality, and fully 30% of all our KO knockout alleles thus far show either partial or complete embryonic lethality. This is roughly similar to what we've seen that was reported previously in the published literature, although I will note that we weren't certain this would be the case as we went into the dark genome, but it has remained to be, this hit rate has remained steady over the course of the program. We recognized fairly early on that these lethal lines, either subviable or lethal lines, are partial or complete lethal lines, were potentially a huge source of important information. At the time, the screen comprised simply of at the beginning, the screen was comprised entirely of a simple assessment of viability, where complete lethal lines were defined as no homozygotes seen at the wean age, while those were defined as partially lethal or subviable in our definition, all those that were less than half the expected number of homozygotes. And as you can see here, I mentioned there are about 38%. You can see this right here. Thus far, we've found over 1,000 genes that are completely lethal as a homozygote. And what's really interesting in this group, which I don't have time to go into today, are what we call partially lethals. Fully 13% of the genome appears to be partially lethal by this definition. And we've developed alternative pipelines specifically for those mutants. And I will note, and I didn't have this earlier, that all lethal lines, we developed an embryonic phenotyping pipeline that I'll describe in a moment, but in all cases, heterozygous mice that were from lines that are proven lethal were run through the adult pipeline that I just described. As I mentioned at the beginning, we actually didn't have an embryonic phenotyping component to this, and we realized that this was a major gap, and so we built one. Working with colleagues around the KOMP2 consortium in particular, we came together and developed a standardized pipeline that allowed us to examine embryonic lethality at different stages of gestation, and in order to time the point of lethality, that would be developing our sort of genotype ratio information. But on top of that, we added an element of assessment and for specific parameters and protocols around gross morphology for each of these time points. And as you can see here in this, these beautiful images from Logan Su and Mary Dickinson at Baylor, we can do micro-CT or high-resolution 3D imaging to develop very rich data sets at all of these phases of embryogenesis. And in the next slide, just to illustrate one of the approaches we're currently taking to analyze these mutants, we can use automated methods to look at the volumetric differences between knockouts and wild types using a segmented atlas developed by Michael Wong and Mark Henkelman's labs a number of years ago. And that this is a great way to get as much deep, rich information out of every mutant line in an automated fashion. So this allows us to move much faster and to identify regions that are different than others. So where do all of these data live? They all live at our IMPC web portal, which is mousepphenotype.org. Very much encourage all of you to go. Not only can you find all of the data, one of the things that might be of particular value to you is to register interest in a given gene. I'll say a lot of genes have been registered, but if we get multiple genes registered, multiple hits for registrations, that really ups the profile for that gene to move into our production pipeline. Although I will say at the end here, we're eager to hear directly from Kids First participants on genes that they'd like to see in the null pipeline. From this portal, you can search by gene, you can search by phenotype, you can search in a number of different ways. If you search by gene, here's an example of the summary page that you can find for phenotype data. You can see here for CIB2, up in the top panel, there's a quick sort of illustration of the different tests for which we either have significant or not significant values, or in some cases for which the tests have not been completed. As I mentioned, these data are released really immediately upon data generation. So in some cases, tests haven't been completed. You can even get pre-QC data by clicking on the heat map table button, which takes you to really a snapshot of the data even before it's been QC confirmed, in case you're really eager for some information. Down below, however, are our final calls on different phenotypes for a given gene. And so you can see we use mammalian phenotype terms, so these data are quite easily searchable, and you can click on the little data bar graph over on the right if you want to see individual data points. And so this is an example of the viewer of those data. Here you can see examples of the mutants and controls. You can see for any given mutant line we have a huge number of controls, so our power for detection is quite high, and this is just an example for the way the data looks is presented to you. We also have developed, our colleagues at the Data Coordination Center have developed image viewers that allow you to visualize both mutants and controls in the same screen. This is just a skeletal, this is an image of our x-rays, so you can look for skeletal abnormalities, but the same viewer technology is available for any of the image data that we generate, including all of the embryo image data that I mentioned before. How do you obtain it? What's nice about the portal is it makes it really easy to navigate your way to animal resources yourself. Not only are the mice and the alleles that are available described in a list, there's a direct button that leads you to ordering information from the repository that holds them, and you can even get detailed information about the individual alleles themselves. So shifting gears slightly, you know, it's been clear to us that the value of the KOMP2 program, it can be amplified by building partnerships with individual groups that are eager to tap into both our expertise, our data, but also the availability of capacity in our individual pipelines to produce mouse models. And so these are just a few examples of the types of partnerships either KOMP2 is built as a group, and I'm going to highlight the Center for Mendelian Genomics because it was really the entree into this conversation with Kids First. So it was an obvious fit for us to begin making mouse models for the CMG, and so we've already built an effective relationship with them, and we hope to replicate that, at least in spirit, maybe not in exact details, with Kids First. And as you can see, all the individual centers have built additional collaborations with either rare disease groups or other or large-scale research efforts, both nationally and around the globe, to continue to take advantage of the resource that we have in KOMP2. So in the final couple minutes, what I thought I'd do is describe the types of opportunities that I see for collaboration between KOMP2 and Kids First. So the obvious and simplest one, and the one at which our CMG collaboration began, is to simply prioritize and nominate null alleles for genes that are discovered in the Kids First program. These are, I would presume, genes for which you have mutations that you would like to know more about, that you think should be made into a mouse model. It's very simple for us. We simply select genes that have a clear mouse ortholog. That is a simple, necessary first step. We, as a consortium for our null allele production, prioritize gene knockouts that are novel. And this makes a lot of sense. You probably already know if there's an existing mouse model. So from the Kids First perspective, it's probably not as interesting to have us make it. And it is part of our mission to cast our net as widely as we can into the unannotated genome by not repeating what's already been done in the community when possible. And then we report back on these existing IMPC models, which perhaps are in progress, and assignments, et cetera. And then we could assign those genes to our pipeline immediately. There's not really any particular challenge in that process. So this is something we can do almost automatically, and so we're eager to start that process with you. But I think the part that might be most interesting to this conversation is the modeling of precision variants. And so we'd like to approach this in a few ways, and I look forward to the discussion around this. I think you're going to hear a little bit more about a framework for nominating clinically relevant disease variants for modeling. We rely on you, on our clinical collaborators, to help guide us towards the most important targets and the most important genes and variants that we could be generating mutants for, and so it's really important we hear from you on that. I think it's always important in every collaboration to define the reporting milestones and timelines. I don't want to underestimate this, because I think it's the way to build a good, useful collaborative relationship is to make sure everybody understands what we can do and what our constraints are, and to have a system by which we keep you up to date with what we're doing. So then we would receive those nominations from the Kids First team for production, and that would entail more than just saying, yes, thanks, we'll go forth. It would require a similar type of exercise as I mentioned above for the null alleles, although in this case we won't use pre-existing knockouts as a filter because we realize these variants probably have different both phenotypic outcomes. And so, but we do need to do a quick orthology search, look for homology, make sure we can make an effective variant that replicates what we see in the human case. And so that's an exercise of back and forth discussion. We would also like to discuss with you potential custom phenotyping packages that are suited to your disease of interest. And I think, for example, within the embryonic phenotyping pipeline, I think for many of the diseases in the Kids First program, this might be the focus of your interest here. And then finally, establish guidelines for data sharing and publication. I think that's an important part of any collaboration to make sure everybody's on the same page there. And so I will say that through supplemental funding from the Common Fund, JAX and the DTCC currently have funding to take your variants and to make those models right away. So we're really eager to get started. And I know BASH, our colleagues at Baylor, also have supplemental funding to make precision variants as well. So I'll just sum up there. And thank all of my colleagues, not only in the KOMP2 consortium, but also throughout the entire IMPC. I mentioned this is both a national and a worldwide consortium, and we really lean on each other to really make progress on this audacious goal, and we are willing, eager, and able to work with our new partners, and we look forward to working with you. And with that, I think I'm done.
Okay. Thanks, Steve. I'm taking the control back.
Great.
And turning it over to James Coulomb.
Hello, this is James Coulomb. I'm the coordinator of the working group for the Kids First, Gabriella Miller Kids First Pediatric Research Program. And I'm first going to give you a very brief overview of Kids First and our progress. And then we're going to talk about how to nominate variants for this unique opportunity. So first off, let's see, I've got to hit that. Right. Okay. Okay. So Kids First is a congressionally mandated program, and our vision statement is up there. I want to emphasize here that collaborative research and data sharing is really key to this effort to foster pediatric research. We have been operationalizing that vision statement by building a data resource that's populated with whole genome sequence and phenotypic data from childhood cancer and structural birth defects patients, and often with their unaffected parents. So it's early days in the program, and it's a long pipeline, but we have 5 data sets that are available for wide sharing, as well as a number of others in the pipeline. And let's see, oh, wait a minute, what happened there? Okay, but what's key here is that as of last Monday, the 10th, the data portal is live, and these data sets can be accessed by people with the appropriate approvals. For you to learn how to access the data portal, there will be I can't even read this now. On Wednesday, the 26th coming up, there will be a portal webinar where the folks from the Kids First data resource will basically demonstrate how to access the portal and how to access the data. Let's see. The pipeline is considerably larger than what's currently available, and we have a number of conditions. We've got greater than 6,000 affected children cases, and most of these are trios, so there's over 18,000 genomes that will soon be in the portal available for analysis. This year, we just recently added a number of cohorts to that pipeline, and we'll have another 8,000 or so genomes coming into the portal, representing a large variety of different childhood conditions. So we're really actually quite thrilled about that progress. We're also very thrilled, although it's early days, we're very thrilled about this opportunity, and it's essentially a pilot to nominate variants for KOMP to make knockouts of these variants. This is an exciting opportunity that we really didn't expect to come along quite so soon, but we want to use this valuable opportunity to pilot this partnership with KOMP. So the goal is to develop mouse strains for studying both coding and non-coding variants that have been identified from Kids First whole genome data sets. And we're inviting the community to nominate variants that have been identified through analysis of Kids First data sets. Let's see. We're rushing this through in a way because we want to get some possible variant candidates into the pipeline for KOMP to work on. We will be therefore nominating the variants that are proposed. We'll be evaluating those nominations with a administrative subcommittee of NIH staff from a variety of institutes that are, these are program officials largely that are involved in the kids working group, and we will prioritize those variants based on the strength and breadth of the supporting evidence that you provide us. We'll then have a dialogue with KOMP2 staff about production of the variants, and the process as so far is hopefully going to be relatively brief and easy. We want you to give us a small description, 3 pages or less, per each nominated variant, and address these items: what data sets you used, what variants you're proposing. Obviously, you need to tell us the phenotype of the human cases that are associated with that variant, what you'd predict that might be a mouse phenotype. That would also feed into the decisions about what possible custom phenotyping services you might want to see. And very important, the supporting evidence for that this variant is really associated with the phenotype, as well as anything else you think is important to tell us, because we're going to try and make these decisions in a very, for NIH, speed of light version of review, so that we can get this, these phenotypes into the pipeline, at least some of the variants. Let's see. If you're proposing a null variant, that's just fine, but we will basically be feeding these to KOMP so that they can put those into their existing decision-making pipeline. And also, there's a email address that you can ask the more technical questions of KOMP personnel about the pipeline and existing knockout or null or knockout models. Again, in line with our trying to get this pipeline primed, we're going to have an initial deadline by close of business on October 26th. We know that's very rapid. We anticipate that there will be additional deadlines, but we need to see what comes in, and this is a pilot, so we need to see what comes in. we may have to come back to you for additional information, and hopefully you'll be receptive to that. We're going to prioritize a list. We'll keep that list, and when we have additional nominations, we won't forget about what was in that previous list, and hopefully be able to prime this pipeline with the best possible variants that we can. We also, again, this is a pilot, and it's a pilot, we hope, for future opportunities with NIH funding to make animal models of the variants that are showing up from these human variants from Kids First data. So we're going to be asking you for feedback at some point of what you think about the process and how we can improve it, because I'm sure we'll be able to improve it. And I'm sure you will have something to say about what we should have considered, et cetera, et cetera. But we're trying our best to make this a valuable opportunity, and we think it is a valuable opportunity that we're really quite excited about. So for questions, you can, again, to technical questions, you can send to the Kids First KOMP email address here. And now I think we're going to open it up to questions, any questions you have at the moment. We have both KOMP staff and people from Kids First here to answer your questions.
So you can message me through the WebEx chat function if that's the easiest way to send a question, or you can unmute yourself.
Hello, this is John Seidman. Thank you for the presentation. I didn't quite get the timeline. So after a mutation is selected for targeting, roughly how long does it take to have that go through your pipeline?
Are you talking about the production pipeline or the decision pipeline?
No, the production pipeline.
Yeah, that's KOMP.
Right. So yeah, there's a couple. So the first thing is it can be typically what we've seen with our collaboration with the CMG is that for a null allele, we can move these into our, and I'll let my other colleagues comment on this as well, but within 1 to 2 months, and at worst 3 months, we can get it into our pipeline. This is mostly because we order reagents in batches, and so we're kind of committed to a certain immediate or existing production commitments. But in general, it's 1 to 3 months, at worst 3 months, but typically 1 to 2 months, we can have a null allele begin the production process. Typically from there, it's really a matter of a few months before we have mice on the ground and we can confirm that we have animals. And then I would say typically for beginning of, or to phenotyping, it's about a year total from concept to phenotype data being complete for the most part. I'd say that's a pretty tight window, but it is the normal window. Now, one of the things I wanted to mention, and I didn't have time in the presentation because I wanted to keep it discrete in an overview, is that this is why we want to hear about custom phenotyping pipelines and goals, because we can accelerate this process. We've been piloting techniques that allow us to immediately analyze the CRISPR-engineered embryos, and it seems to be effective in some cases. That may be of interest to individuals, to people. There may be phenotypes we may expect to observe perinatally, and so we could structure our pipeline to do that observation first. So I wanted to make sure you understood that the standard pipeline will take about that much time, but the potential custom pipelines could be more rapid.
So just for a follow-up question, so obviously I think many of the groups that are participating in GMKF have thought about mouse models for other genes that are associated with the condition that they study, so they've actually developed phenotyping pipelines for the specific condition that they're looking at. And I just wondered, so when would mice become available as heterozygotes at some stage during the course of that year, or do we wait to the end, or how does that work?
Yeah, so I can speak for JAX, but I'll let others pipe in. We make our mouse available in terms of visibility, and you can place a register interest and place an order as soon as we have genotype confirmation, which is the QC assay after we get genotype transmission. We allow distribution once the colony is expanded up, which is 1 to 2 generations, depending on how many animals transmit the allele, but we make them available prior to phenotyping. And in fact, historically, most of our orders have come prior to phenotyping.
Okay, thank you.
And this is from TCP, and the process is very similar here. As soon as we know that we have the desired allele transmitted and the allele in the mouse line itself past QC, we make the line available. And generally, it can be live mice can be sent. It depends on the numbers of mice, but we often have live mice available for sending after the first back cross of those QC passed animals, simply because we generally make more than we need for our own breeding in that first round.
All right, I'll just chime in further. This is John at Baylor. I think echoing Steve and Laurel's sentiments, we'd expect to be able to rapidly get nulls into our allele. And addressing the question about non-comp sites where they might already be set up for performing systematic phenotyping, although our normal thinking on distribution of live lines is about transmitting, enough to establish a small breeding colony at a recipient investigator site. If there were circumstances where individuals might want to receive essentially an experimental cohort, this would certainly be case by case, and it might require funding support to expand our breeding exercise, but that's certainly something that could be considered on a case by case basis.
I received a question through the chat from Simeon Boyd. Will the mouse phenotype data be reported to the PI, and will there be an embargo period before that data is publicly available?
I think that's a great question. For null alleles, because it's part of our standard remit for comp, I think we would send them through without embargo. However, for specific variants, that is sort of the topic of this discussion. That is something, it's kind of why I put in my bullet points, we need to discuss that relationship. We do understand that there might be interest in an embargo that fits with the publication policy that we would put together. So to be determined, I think but I think we should discuss that. I think it's an important point. The nulls were committed to rapid release of those data.
Yeah, this is an issue that we would need to discuss amongst the Kids First leadership and working group as well as with COMP. We're putting this program together very rapidly, so a lot of details have not been worked out yet.
Yeah. So that's on the radar.
But that is on the radar.
Yeah, this is Laurel again from TCP, and I'm not suggesting that this is what we want to do here, but I just wanted to explain my experience. We had a similar collaboration with a group called Care for Rare here when we were funded on a previous program funded by Canada. And what we had agreed with Care for Rare is essentially a 6-month embargo on releasing data or the allele information publicly. So I think in this case it will really depend on not only what Kids First wants, but also the Common Fund and the NHGRI who are funding the supplement may have strong feelings one way or the other about embargoing data. But I do think it's something that's entirely very important to discuss and make sure we're all on the same page.
Hi, can I this is Terry. This is Terry at EBI. We're part of the Data Coordination Center for the IMPC. We're also funded by the COMP mechanism, and it falls in my team's remit to do the tracking of all the mice across the whole consortia to reduce duplication and coordinate across the centers. And I'm just wondering, James, you said this is going at light speed. would Kids First consider the variants that the KOMP centers would make for this program patient sensitive, and that you would not want that sequence information publicly available and have to be treated via access by a data access committee?
No, I don't believe so.
Sorry, I had you muted. Say it again.
Sorry?
Say it again. I had you muted.
Oh, you had me muted. No, I don't believe so. We would probably, though, have to consult with the members of the working group who are real data sharing experts to make sure of that answer. But my impression would be no, that wouldn't be.
So we'd like to be part of that discussion because we've been in a completely open embargo attitude with the mouse tracking, and if we involve KOMP resources going forward, we'd like to be part of the discussion to figure out how to best handle that.
That's great.
Great. Well, I'll follow up email. Thanks.
Yeah.
This is Austin. I'm curious. You've talked a lot about knockouts. Are you accepting proposals for introduction of missense variants?
Yes,
so,
very much. Oh, go ahead.
Yeah. Sorry, yes, I mean, if the knockouts come in through the Kids First nomination process, again, we will pass them on to KOMP with our recommendations, as it were, because it's important to us that Kids First data gets used, and this is one way of demonstrating that use. So we would probably pass them on with our gold seal of approval if we think it's reasonable.
And I just want to make sure I heard the question as well. Was it for structural variants, did you say?
It's either choosing structural variants as well as missense variants within a particular gene.
Yeah, we are interested in both of those. I think those are, and as I said, that these are the more special cases. This is what we have the pilot funding to do in addition to developing the workflow, as you hear around publication policies. That's all part of it. But both structural and missense variants are part of the goal.
Great.
I think we answered John Seiden's question about missense variants as well. Another question from Simeon Boyd. Are there plans for knock-in models or just CRISPR-based exon deletion?
Knock-in meaning missense type knock-ins? Certainly yes. I would imagine in all cases there will be still CRISPR-based because I think we only use that approach now. But certainly, yeah, I guess I didn't emphasize it enough. Missense, structural variants are all part of the discussion. These are the things that we don't currently do, but we would like to do as part of this pilot.
Any other questions?
Although I misspoke there. We don't currently do as part of KOMP. Every single center does this a lot as part of other collaborations that they have, core services that they provide, et cetera. But missense and structural are not currently essential part of KOMP, but we would like to include it.
I guess if Simon could just clarify that by knock-in he did mean missense mutations. If he meant something else, it would be good to have that clarification so we can talk about that a bit as well.
He said yes. Okay. Any other questions?
Just a quick question on the knock-in and or structural variation. What about duplication events or those kinds of larger events are also of interest?
I would say yes, they are of interest. I think we would need to discuss them and think about, depending on what they look like in terms of devising the engineering strategy. Some are much harder than others, and so we would want to have that discussion. But again, this is a pilot to see not only what we can do at scale in terms of other types of engineering, but also what suits our phenotyping capabilities well. So we are eager to discuss those. So certainly they would be part of the discussion.
I received another question in the chat. What is the timeline for the mice? Will they be made available to others, and on what timeline?
Yeah, so as I mentioned before, null alleles, we have a lot of experience with this, and null alleles are made available, as we discussed before, basically upon QC confirmation. And then, as you heard before, I would say, in addition, the same discussion about embargo that James was speaking of before, that we need to have as a group, both with NIH group and as collaborators, is what, for the precision and structural variants, how that embargo would look. And I think that discussion would include both mouse models and the data that would come out of it. So I would like to wrap that into the same discussion.
We agree.
Agreed.
Everybody's nodding their head in here.
Yeah. So just for those who don't really know the timeline, so when we say as soon as QC is available, so basically at TCP, and I don't want to speak for John and Steve and Kent necessarily, but at TCP it generally takes probably 4 to 6 months to have mice available for distribution. So that's not saying we have the phenotyping done. That's saying we might have a couple of extra mice that we could share with the person who nominated the gene so they could start breeding their own colony. For larger numbers of mice and for making up the phenotyping cohort, it takes a bit longer than that. But just for those who aren't really familiar with the timelines involved in model production, and this 4 to 6 month window is assuming that everything goes well, it works the first time we try it, and that sort of thing, and that there's not any technical glitches along the way. And while missense mutations and smaller structural variants are fairly straightforward and fairly routine in most centers, things like larger duplications and inversions are much more towards the technical development side rather than the ready for prime time scale production side, at least at TCP. And I don't want to speak necessarily for Jackson, Baylor, and UC Davis, but we have had some experience with larger duplications and inversions, but they certainly aren't at the efficiency level that we see with the missense and smaller structural variants and nulls.
Yeah, I will echo that exactly. 4 to 6 months is our typical range. It can be slightly earlier for a null on some occasions, but that's typically when they're available for distribution here. And I would also echo that for structural, that's why I mentioned it should be, these would have to be sort of custom discussions, because while precision missense variants can be generated quite routinely, others maybe not so routinely.
One last question from Simeon Boyd about whether non-coding variants are included, and I believe the answer is yes. This was already answered.
Yeah, we would, again, as this pilot, we're really looking to explore all of these things and ways in which we can use our production capabilities and phenotyping pipelines. So, you know, those are definitely on the horizon for the KOMP2 program.
And if I can just add to when we're talking about non-coding variants, it's fairly easy for us to identify orthologous mutations in mouse for coding regions. It's a little more difficult, and I certainly don't have a high level of expertise in this, to identify synonymous or non-synonymous non-coding regions. So there would have to, as Steve mentioned, for other types of alleles, be some discussions going back and forth that really try to explore whether, you know, the orthologous or homologous region actually existed in mice and what kind of changes might be needed in order to give a best chance of success in terms of modeling that non-coding variant.
Thank you.
Just one more quick question. You know, as was highlighted, the Kids First is also interested in the cancer side. Is cancer at all in the portfolio of phenotyping that we should be considering, or no?
So if I might jump in and answer that, this is Laurel at TCP. We have, I guess, 2 assays that might specifically address cancer. One is histopathology. So TCP is fortunate that we have board-certified mouse pathologists on our team. So we do a complete histopathology on 40 tissues, or 41, I think, if they're female. at the end of our pipelines. So there, any sort of precancerous lesions or tumors would be identified. The other assay we do that is relevant to cancers, we do something called a micronuclei assay, which is a sort of a high throughput flow cytometric test looking for genomic instability. So those are the two assays I think that are most relevant to cancer that are in our current pipelines. And I don't know, I won't speak for Jax or Baylor about what they might have at their sites.
Both of those are tests that we don't currently routinely do for everything, but we have done in the past, or we've piloted, so we could easily work them in if they would be of interest for a cancer-relevant variant. I think that's a good discussion. It would be good to hear these are pediatric cancers, so age of onset may not be the challenge it is for other cancer models. So I would like to continue to discuss it because I think it would be really important to try to model those as well.
And I think that's something that we might, in particular, focus this particular discovery partnership on in terms of future rounds of funding and production and phenotyping with an explicitly cancer-driven focus.
I would say that the Kids First program is certainly very interested in those variants.
Right. The nominations that will get the first administrative review from us certainly is open to cancer applicants and cancer-relevant variants as well. It is past 4 o'clock, so thank you to everyone. I do want to kind of commandeer just a couple more minutes for whoever has time, just to orient you to the upcoming events. And as James mentioned, the Kids First Data Resource Center has launched their portal very recently, and we are hosting with them a full-day webinar on September 26th. In this slide deck is a registration link. It's also available on our listserv. We will post these slides on our Kids First Common Sense website so that it's available to you. We also, we have been invited, and we are all inviting you to observe and watch KOMP2 IMPC's upcoming collaboration day of their annual meeting. We will announce this information on our listserv, so anybody who's on right now is welcome to join that as well. And Kids First is hosting a poster session one evening of the American Society of Human Genetics meeting. Several members of the KOMP team will be there. I think this is going to be a great opportunity for some of our Kids First investigators to start talking to the KOMP team about this specific opportunity. But it's also just a great chance to see what's going on with the Data Resource Center and each of our XL1 cohort projects. Thank you. All of this will be available online.